Okay, because we have very tight time for each speaker, 20 minutes, we are going to start these other 4 genetic related topics. It's a pleasure for me to present the Dr. Trevor Walker, managing adaptability in a breeding program while balancing gain and diversity. So, thank you. Go ahead, please.
Yeah, thank you very much. Thank you to the conference host. And also, big thanks to the organization that I work for, the Tree Improvement Program. You'll see the logo here that funds this research and uses this research. So our program, the Tree Improvement Program, is a recurrent selection program. We use artificial selection and plant breeding methods to make genetic gain. Starts with picking trees that have good phenotypes, making crosses among the trees that we think have good genotypes based on prior knowledge to try to create new combinations of genes that have all the good genes from their parents and none of the bad genes. And it involves rigorously testing those genotypes so that we know which trees are good parents for going out to the seed orchards, and for trying to find that those trees that have the best combination of good alleles, the best sets of good alleles, to repeat the process over again. And we select those and put them back into the breeding program. And right now we're in our fifth time around this circle. Planted our first 5th cycle tests this past winter. And our second, sometimes we've gone backwards. Every breeding program has baggage and things that come along with it. But always looking forward. I don't know how widespread this term is, the breeder's dilemma, because when I Google it, I get a lot of dog breeding responses instead of like population genetics, plant breeding responses. But the way it was taught to me is the breeder's dilemma is that diversity in your population is at direct odds with short-term gain. Diversity is necessary to continually have opportunity to break apart those good alleles in a population from the bad alleles. It's the number of good alleles that you can start off with. But also you need diversity to buffer against things like climate change, if environments change, or co-evolving pests, and also co-evolving markets. For example, if international markets change in such a way that a trait like the density of the wood becomes important, you have to have a diverse population to answer that. And like I said, and I'll repeat it because it's what a lot of this talk is about. As you increase the diversity of your population, you're dropping that average farther and farther down. So here's an example of the gain that we've made. I've got the percent improvement in volume as the x-axis right here for one of our populations, and then this dotted line is the wild, non-improved, the way that before any artificial selection. And so after our second cycle, I don't have the data for the first cycle, but with our second cycle, this is actually the parents of the second cycle. Well, we made selections from some of those parents, and those trees were carried on to our third cycle. When we look at all the third cycle parents, their breeding values here, we see that on average we've made gain. And we continue to make selections from those. Those were the parents of our 4th cycle selections. We're testing those right now, and we've already started to make some selections for those. So increasing the gain is important, and diversity is really important too. So each one of these ramettes, or each one of these trees will have a 5th cycle selection top grafted onto it. And it's a lot of work to drive the lift around and breed all those things. So we want to make sure that we're getting the bang for our buck. If we look at some diversity statistics that go along with that gain, with our third cycle, we had 703 trees participating in that breeding effort. The third cycle was interrupted with the 2008 economic crisis, and then also changes in land ownership in the southern United States. But we got our act together. And in the 4th cycle, we had almost 1,000 parents participating in that breeding. The 5th cycle is in progress, but our projections over the next 5 years or so is to have 728 individuals participating. Of course, that's only one part of the picture. There's a lot of relatedness in these numbers. If you look at the number of founders, presumably unrelated individuals that are represented by those numbers, we've been dropping that number down as we make among family selection and get rid of trees that we just don't think are contributing. We're increasing the average relatedness between any two individuals in the population. And then I really like this status number that Dag Lindgren proposed. It represents, it's an effective population size measure. And this number really scares me right here because it didn't drop near as much between the third and fourth, but as a tree breeder, a number like that is not what I'm used to seeing. But it is, what I will tell you is that I know theoretically this number always needs to be high enough so that we can make enough parents for an open pollinated seed orchard. That's my rule of thumb for that. So we have got this complex pedigree, that's what this is, founders on the top, and then the next generation. And you can't just pick two trees from your pollen inventory and flower inventory and decide to mate them because you got to trace it all the way back to get that progeny breeding. So we use a software that was proposed by Brian Kinghorn. It's been around for some time and widely adopted in the livestock programs, but also aquaculture and plant breeding programs. It's very well suited for managing the balance between gain and diversity with trees. And the way that this algorithm is set up is, first off, you tell it two things. One is how many crosses you want to make, how much work you can do, which you should always do as many crosses as you possibly can. And then you tell it this thing called the target degrees. And then based on your breeding candidates, you've got an x-axis here. Oh, I knew I was going to hit that. You have an x-axis here that is the mean relatedness among those. And then you have a y-axis that is the genetic worth of the progeny. So as you increase as you decrease the diversity in your breeding plan, you're going to get more gain. And that's what this black line represents is the most gain that you can get, is that black semicircle, the most gain that you can get for a given amount of relatedness. And what you tell the software is that the angle of this green line, obviously you want to get as much gain as you can from the trees that you have. You just need to conceptualize that green line. So if you have a green line at 0 degrees, that means just maximize gain. It means take number 1 and cross it with number 2. But you've told it you need to do, say, 150 crosses or 200 crosses, so it's going to involve more individuals than that. But target degrees of 90 means don't put any emphasis on gain. Get the most diverse batch that you can get. And a common number is 25 degrees. Foresters tend to be a conservative bunch. We've been using 35 or 45 degrees. But what you have to do is you run this thing, and this is just a animation showing how the solver, like an operations research solver, is cycling through a list of crosses to try to get as close to that black line as it can. Until it takes about an hour or so for most of our runs to reach the solution. So I've talked about managing gain and diversity, and it's never that simple. We also have to keep up with adaptability. So breeding zones are how adaptability has been traditionally kept up with, and they're typically defined with common garden trials or provenance tests. And so the pine seed source study that was put in by the United States Forest Service and summarized really well by Ron Schmittling in this publication is where we got most of our guidelines. And it wasn't a breeding zone solution that we started off, or that we've really been operating on. It's been this observation that as you move seed from coastal origins more inland, you tend to get improved growth up to about 5 degrees Fahrenheit, where you have a risk of cold damage. So I didn't explain this graph, but that's what it means. We have our own provenance trials where we've seen the same thing. Oh, let me explain this. So this is an example of a common garden study where the circle is where we've collected seed from a mother tree, and then every square we've planted that common garden trial. Huge effort that the co-op did in the 90s. And we saw the results of that on this axis here. We have the provenance source in degrees Celsius. And then on this axis here, we have where it was planted in degrees Celsius. And the coastal warm source material does better on average. That's what this plane is, until you push it too far. And then the more cold tolerant stuff does better. And this is a really good paper. I copied the methods off of this that Nick did using the factor analytic models that Allison presented a couple days ago. And I'm going to blow through it in the effort of time. But we followed those methods. We calculated this matrix. If you recognize it, that's great. At the end of the day, we were able to define 3 environments where the performance of trees were correlated were well correlated with each other. And what this says is whenever we look at our different genotypes planted in this orange region, we get a similar correlation. But once we go to the purple region, that correlation is not as stable, and a different ranking of genotypes appears. And so these 3 areas we have been using as breeding zones. As with most programs, we started off with mini breeding zones where we've tried to maintain adaptability, and we've reduced that as we've gotten numbers. And for the 3rd cycle and the 4th cycle, based on those things that I showed you, we've said we need to have 3 breeding zones to maintain adaptability. And what the implications of that are, when we're balancing gain and diversity, we don't allow matings for trees that are in different breeding zones. You're only allowed to mate within your breeding zone. And that seems pretty arbitrary. It's not arbitrary, but right, that's like a hard and fast rule. We really need to evaluate it. It's simple, it's effective, but certainly it's suboptimal because, right, the coldest source Piedmont tree is probably, the coldest source coastal tree would probably make a great cross with the warmest source coastal tree. It ignores any co-ancestry that's built up among the populations, and it's always also not straightforward. So what we've done instead is we've been looking at the winter temperature source origins of our population. So here's a distribution of all our 5th cycle selections, and in Fahrenheit, 5 to 25 degrees is the max. And what we're trying to do is we're trying to put that across the landscape, make sure it matches the deployment area. It's important, this is super busy, but it's important because the mean temperature of the source origin has a negative correlation with volume, and then this index breeding value, which combines volume, straightness, and rust, it has a negative correlation with that. The colder trees grow slower. So I looked at 3 different scenarios. 1, a baseline running mate cell without any constraints, and then number 2, using rigid breeding zones, and then finally using what I'm calling flexible breeding zones. And the way the flexible breeding zone works is that I divided this thing into 5 degree bins, and I put in there's an input into the software, and what this matrix says is you're allowed to make crosses among adjacent bins. In fact, in the coastal region, you can skip 2 bins, but avoid it. You're not allowed to make crosses among wide crosses. This is the target number of crosses we're going to do. These sum up to 500, and this is the weight we're going to put on it, 0.7. These are the results. Okay, so no constraints. It just picks the best trees to mate. Breeding zones, you see these clear, inefficient blocks that are going to represent some loss of optimality. And then with the flexible zones, you see some crosses being made. Sorry, I didn't explain this graph. This is the pollen winter temperature source, and this is the flower, the female winter temperature source. So the 2 solutions are similar. They look similar with how the crossing is done, and they look similar if you look at the crossing winter temperatures, the output from those. Obviously, the no constraint only chooses to breed warmer source trees, but the breeding zones and the flexible zones have similar output here. And you really see differences when you look at the gain in diversity. So red is no constraints. That's not acceptable. Green being the breeding zones, this is how much gain we lose because of that constraint. The blue flexible zones is a lot closer to the no constraints option. And it's also intermediate when it comes to diversity. And so I'm going to go back to this because it says I have 1 minute. Yeah, so I just did this for one generation just to test it out. This isn't a rigorous analysis. But that's what I like about this software and this algorithm is that you can run a couple different scenarios, maybe change that target degrees, look at the difference, look at how much gain you're leaving on the table. Recently, we went from 45 to 35 because of two things. There was so much gain we're leaving on the table. And then when you look at this table right here that shows the number of parents that were used, for the no constraints, only 126 parents were used. For the breeding zones, a lot of parents getting used. And for the flexible zones, somewhere intermediate, this software is really great for looking at the impacts on gain and diversity. So, yeah, with that, I'll take any questions if there's time.
We have time. 2 minutes of questions. Yes, go ahead.
Hey, Trevor. Cool to see you're doing lots of the same things we're doing. Do you find as your breeding deployment zones change, you're updating values and rules to who can be deployed where? Are you backcasting that onto existing orchards or just redesigning them for the future?
So one thing about the southeast United States is kind of like the wild, wild south. There's no regulation on what you plant anywhere. Rather, what we do is we print these maps that show, they look just like when you buy a bag of seed for your garden, for your tomatoes, that say when you should plant. We just do these plant hardiness maps. And green means very low risk, yellow, slow down, and then red means high risk of cold damage. So that's the approach that we've taken. So far, we haven't really had any sort of proactive changes in those things. And we're using old data to make those calls. But I will say this, every year we plant progeny tests. And so, right, you have to plant trees to know how climate is going to affect those. So we do have that in our back pocket.
We have time for one question more.
Okay, so do you actually have orchards or are they crosses you're deploying?
Yeah, so both. And I'm sorry if I didn't do a good job of explaining that. We have breeding orchards, which maintain the breeding population. And then once a superior parent has been identified, our members are eager to graft that into their seed orchards and plant that seed across the landscape. So it's the developmental population I've been describing.
Yeah, so just to clarify Trevor's question, it's not an orchard seed lot, it's from two parents.
Yeah, these are all controlled crosses.
Okay, sorry, there is no time for more questions. Thank you very much. The next presentation is Theodora Lo.
Hi.
Assembly and annotation of the black spruce
The
genome
remote?
provide insights on spruce
How do you use this?
phylogeny and
This is the
evolution of stress response. So,
Great.
go ahead.
Thank you.
Thank you.
Yeah, so hi everybody. I'm Theodora, a master's student in Dr. Nanj Burrell's lab at Canada's Michael Smith Genome Sciences Center. So yeah, my talk is definitely a little bit different than the other topics here at this conference, but this is not the topics session, so I guess it's quite fitting. Yeah, so today I'll be presenting the assembly and annotation of the black spruce genome and how analysis utilizing this new genomic resource has provided insights on spruce phylogeny and evolution of stress response. Yes, so boreal forests have been considered a significant carbon sink, and black spruce, being a dominant species in Canada's boreal forests, are a major contributor to that. It's also an economically important species, being highly valued for its wood products, particularly in eastern Canada. Now, although black spruce has been historically resilient to fires and relatively resistant to some pests that other spruce are susceptible to, notably the spruce budworm, recent climate extremes have increased the severity of abiotic and biotic stresses, and as a result, declines in black spruce growth have been observed. So nowadays, studies are more focused on identifying adaptive traits and understanding the genomic architecture underlying these traits to guide the development of assisted breeding and migration strategies, for which genomic resources play a critical role in. That being said, there are a few resources available for black spruce, and those that are available are mostly transcriptome related. So due to the lack of genomic resources for black spruce and just other spruce in general, phylogenetic studies typically use mitochondrial and chloroplast DNA, but these previous studies observed differing tree topologies depending on the regions that were used. And this has largely been attributed to ancient reticulate evolution affecting these genomes due to the different modes of inheritance. So specifically, the chloroplast genome is paternally inherited through pollen, which can be disseminated across long distances, facilitating ancient reticulate evolution. The mitochondria genome is maternally inherited through seeds, and the nuclear genome is biparentally inherited. And hybridization has been observed within the spruce genus, as exhibited with interior spruce, which is an admixture of white, Engelmann, and Sitka spruce. Black spruce also exhibits rather unique traits compared to other North American spruces, both morphologically and ecologically. So just as an example, on the bottom left there, you can see a few pictures of the needles and cones of several North American spruces. And you can kind of see that black spruce has shorter stiff needles and smaller, rounder cones compared to the other spruce. And then on the right is a map of Canada with the ranges of the spruces. I do want to point out that although I've only included a map of Canada, that the ranges do extend partially into the US as well. But from this map, you can see that the range of black spruce is relatively distinct compared to the other spruce, with the exception of white spruce. Now, although the range of black and white spruce are largely sympatric, the two have adapted to rather distinct ecological and climatic niches, with black spruce inhabiting cold, wet, nutrient-poor soils, and white spruce inhabiting warmer, well-drained soils. And besides these species-specific traits, the transcontinental range of black spruce has allowed for adaptive evolution, with some populations exhibiting local adaptation and some populations exhibiting historic differentiation in the standing genetic variation. So further analysis could provide insights on how black spruce has adapted to its rather particular ecological niche. Could I get to the next slide, please? There we go, thank you. Yeah, so that brings me to the research aims of this project. So first, we wanted to develop genomic resources for black spruce. Particularly, we wanted to provide a genome assembly and annotation for the community. Then, utilizing this genome assembly, we wanted to investigate the phylogenetic relationships among several spruce species. And then lastly, we wanted to perform some comparative genomics analysis to hopefully identify genes that are potentially involved in stress response. So recently we completed the genome assembly of the black spruce, and this is now submitted to NCBI, so it's publicly available. This assembly was done by Lauren, who's the assistant bioinformatics coordinator in our lab, and to do this, she used two types of data, short reads and 10x Genomics Chromium linked reads, totaling to a fold coverage of about 46 times. The pipeline that was used for this assembly is shown on the left there. So just very briefly, the reads were first assembled using Abyss, followed by scaffolding with NT-join. And then since we had linked reads, the long-range information in those reads can be used by Tigment to identify misassemblies and cut scaffolds at those positions. Then further scaffolding was performed using ARCs, and gap filling was performed using Sealer, ultimately yielding a final assembly with an NG50 of about 36 kilobases and a reconstruction of about 18 gigabases. So here I just wanted to point out that the genome assembly was done using tools that were all developed by our lab, the VTL lab at the Genome Sciences Center. These tools, as well as a few others that were developed by our lab, were also used in the assembly of a few other spruce genomes, so they're definitely scalable to these larger genome sizes. And if you're interested in checking them out, you can visit the GitHub link, which is at the top right there. Then to annotate the genome, we first masked repeats in the genome assembly using repeat masker. Then to identify gene models, we used a breaker, providing both publicly available protein sequences as well as RNA-seq alignments as evidence, and a pre-trained Augustus model. And then functional annotation was performed using NTAP. So after all of this, a few post-processing steps were performed. In particular, we only kept complete genes in our final annotation. And by complete here, I'm referring to genes that start with a start codon and end with a stop codon. And then we removed any genes that contained PFAM domains that are found in GAG and POL genes, since these are characteristic of LTR retrotransposons, which should have been masked in the repeat masking stage. And then lastly, we flagged gene fragments based on RNA-seq alignments to the genome. So to summarize the assembly and annotation, I'm just going to provide a few statistics for both of those, beginning with the assembly. So 3 common statistics that are used to evaluate the quality of the assembly and or annotation are the NG50, reconstruction, and number of complete BUSCOs. So the NG50 is basically a measure of the contiguity of the assembly, and is more specifically defined as the length at which 50% of the predicted genome size is contained in contigs longer than this length. Basically, the higher this value is, the more contiguous your assembly is. The reconstruction is the sum of all the contig lengths in your assembly. And then BUSCO, this is a measurement of the completeness of your assembly and or annotation based on gene content. And the BUSCOs are a set of single copy orthologs that are highly conserved among related species. So the higher this value is, the more complete your assembly and or annotation is. So going back to our statistics, as I mentioned earlier, we achieved an NG50 of about 36 kilobases and a reconstruction of about 18 gigabases, which is comparable to the estimated genome sizes of other spruces, reported to be about 20 gigabases. Then onto the annotation statistics. So I've just provided a breakdown of the statistics at each stage in the annotation pipeline, but ultimately we ended up annotating about 66,000 complete genes and about 69,000 transcripts. So now that we had our genome assembly, we wanted to investigate the phylogenetic relationships between several spruce species. To do this, we generated phylogenies from nuclear, chloroplast, and mitochondrial genomes of 6 spruce and 2 pine species using MASH-tree. So in the phylogenies that I'm going to show, the values at the nodes are your bootstrap values, and then the branch lengths are going to be proportionate to the number of substitutions per nucleotide position. So starting with our nuclear phylogeny, you can see that our pines and our spruces form 2 distinct clades. You can also see that Sitka spruce is a close sister group to interior white and Engelmann spruce, which is expected since white and Engelmann and Sitka spruce frequently hybridize with each other to form interior spruce. To further illustrate this point, I've just included a diagram from hybridization studies. And you can see that Engelmann, Sitka, and white spruce are connected by thick lines, which indicates that crosses between these 3 species are highly successful. So overall, the topology is reflecting the close proximity of these species ranges and the natural hybridization between these species. Another thing I wanted to point out is that Norway and black spruce are sister groups to the other North American spruces, which reflects the phylogeographic speciation. And this is expected for Norway spruce, given that it's European. And then in the case of black spruce, if we go back to our hybridization diagram there, you can see that black spruce is connected to the 3 North American spruces, Engelmann, Sitka, and white spruce, by finer lines, which indicates that crosses between black spruce and those 3 species have either failed or had low success rate, which indicates evolutionary divergence. Now looking at our mitochondrial and chloroplast phylogenies. So generally speaking, again, black spruce appears as a sister group to the other North American spruces. Looking a bit more closely at our mitochondrial phylogeny first, we see what we kind of expect to see given the geographic distributions of these species. So we see Sitka as a close sister group to white interior and Engelmann spruce, followed by black spruce, and then the European Norway spruce. In contrast, when we look at our chloroplast phylogeny, there's 2 observations that kind of stand out. So there's Sitka spruce at the basal position, and then Norway spruce appears as a sister group to interior white and Engelmann spruce. Although we know that Sitka is closely related to those 3 North American spruces, and yeah, Norway is the European. So because the mitochondria is inherited through seeds, which disseminates across much shorter distances than pollen, which is what the chloroplast is inherited through, the topology that we're seeing in the mitochondrial phylogeny is likely more representative of vertical descent, whereas the conflicts that we observe in the chloroplast phylogeny and nuclear phylogeny regarding the positions of Sitka and Norway spruce are indicative of ancient reticulate evolution. So now switching gears a little bit and moving on to the comparative genomics analyses, we first looked at black spruce specific orthogroups. And to do this, we identified them using OrthoFinder. Now we wanted to get an idea of the functions of the genes in these black spruce specific orthogroups. So we looked at the distribution of the eggnog functional categories and PFAM domains in these genes. And so from this chart, you can see that many of the genes were assigned to the poorly characterized functional category, but there were still many genes involved in cellular processes and signaling and metabolism. So I'm going to talk about these three categories in a bit more detail. So despite being poorly characterized, analyzing the PFAM domains in these genes revealed that many contain the domain of unknown function DUF4283, followed by the EAMA domain and the FBOX domain. So the EAMA gene family is involved in transport, and the FBOX superfamily controls a diverse set of biological processes, including growth, development, and abiotic stress tolerance. Then moving on to the cellular processes and signaling category. So shown on the bottom left there is a bar chart that shows the distribution of the subcategories within each of the supercategories. So looking at cellular processes and signaling, which is in purple, you can see that the most represented subcategory is T. So I decided to highlight a few of the most represented PFAM domains within that particular subcategory. And this included the NB-ARC and LRR1 domains, which are found in NLR proteins, and these have been shown to play a central role in resilience to biotic and abiotic stresses. This also included the lectin-leg-B domain, which has been predominantly identified and characterized in L-type RLKs, and studies have shown their critical role in pathogen response, and there's also been a few studies identifying links to abiotic stress response and developmental processes. Then moving on to the metabolism category, so you can see in blue that the most represented subcategories were G, C, and Q. And among these three, the most represented PFAM domain was the UDP-GT domain, which is found in UDP glycosyl-transferases, and these play a role in cell wall lignification. And this was followed by the P450 domain, which is characteristic of cytochrome P450 oxygenases, and these are involved in protection against both abiotic and biotic threats, including playing important roles in oleoresin defenses of conifers and interactions of conifers with insect pests and pathogens. So besides looking at black spruce specific orthogroups, we also wanted to identify genes that played potentially adaptive roles. So we performed positive selection analysis on the single copy orthogroups identified between the 8 species. And this analysis revealed that 45 genes were positively selected in black spruce, 38 of which could be annotated with functional categories based on hits to the Airport 11 database and or literature search. So among the 38, many of the genes are involved in development, followed by stress response, and then growth. Among the 12 genes involved in development, 2 of them were involved in regulating flowering time, Frigida, which is also involved in temperature adaptation, and WNK1, which regulates flowering time through the photoperiod pathway. The 12 also included a trihelix AA kinase chimera, which is associated with leaf development. 8 genes were involved in stress response, 2 of which are involved in plant immunity and plant immunity regulation. The PBL27 serine threonine kinase, which is involved in resistance against fungal and bacterial infections. And there's also an ATWRKY3 transcription factor, which plays much broader roles in the positive regulation of biotic stress response. Then besides biotic stress tolerance, there's also some genes involved in abiotic stress tolerance, particularly to cold. This included the glycerolipid A1 lipase PLIP2. So in conclusion, the assembly and annotation of the black spruce genome is now publicly available. In particular, the assembly is available on GenBank, and at least for now, the annotation is hosted on Zenovo, but we definitely have plans to submit this to NCBI soon. In our phylogenetic analysis, we observed genetic differentiation between black spruce and the other North American spruces, which reflects ancient divergence of its lineage. We also observed at least 2 signatures of ancient reticulate evolution affecting the spruce chloroplast and nuclear genomes. And then lastly, our comparative genomics analysis identified genes with roles in development and stress response. So yeah, that concludes my presentation. I just wanted to mention that this was done as part of the CanSeq 150 initiative and supported by Spruce Up. I want to thank the funders, Genome Canada, BC, and Quebec, as well as those that contributed to this work, my supervisor, Ananch, Jorg, Ashley, Renee, Lauren, Christina, Jean, and Natalie. So yeah, thank you, and if anyone has any questions, I'll take them now.
Super good in time. So questions? Yes, please.
Thanks very much for your talk. Very nice. Just a quick question about the funder selection analysis. Was that done using camel or something?
Yeah, with CodaMel, yeah.
And then have you looked at whether or not any of those genes you identified have been flagged in any other conifers?
Other conifers. I haven't looked into that yet, but that's definitely something that I can look forward to doing in the future, since I've definitely seen some papers on positive selective genes. So yeah, thank you for mentioning that.
Yes, please.
Oh, thank you. Looking the wrong way. Thank you for your very nice talk. It was very interesting. I used to dabble quite a bit in Picea phylogeny myself in my early days, 30 years ago, using the rather primitive methods of RFLP on chloroplast DNA, and it was exactly like the dendrogram you just presented. But what I wanted to ask is, since you are interested in the commonality of certain genes from black spruce to others, my best bet to look for relatives of black spruce would be, on the one hand, Picea rubens, red spruce,
right.
and then Picea omorica, the Serbian spruce that occurs as a small relic in 4 mountain ranges in Balkans.
Great. So, yeah, thanks. Thanks for that suggestion. Thank you.
Any other question? We have time for one question more. Yes. Sally, please.
Yes, so in your mitochondrial phylogeny, I'm just wondering if you have given any thought to where the samples came from and whether there might have been mitochondrial capture with, with species colonization moving in.
Yeah, that's a good question. So the phylogenies were generated from the assemblies. So yeah, I haven't really thought a lot about that particular question. Was there any interesting observations in that particular phylogeny that you?
What's just been observed in a number tree and other species that if you have one species present and another comes in and hybridizes in the first generation, then even though it might revert to being a relatively pure typic species, that it might carry the, because the seeds are maternally transmitted and the mitochondria rather are maternally transmitted, then they end up kind of resident. What first come first persists, so it can create quite a different pattern. And I'm just looking at your phylogeny there. I just wondered if that was what was going on, because I know that's happened to some extent with the spruces in BC.
I see. Okay. Yeah, I can definitely look into that as well. Thank you so much.
Okay. Thank you very much, Theodora.
Thank you.
So the assembly of white spruce enable improved annotation and generation of a chromosome-scale epigenetic map.
Thank you. Can I skip to the next slide?
This is next. This is for the next and back, and this is the laser.
Okay. Okay. No, thank you. Hello, everyone. Oops. Is it on? Okay. Hello, everyone. My name is Irem. I'm a Master of Bioinformatics student at UBC in Dr. Inanç Boral's lab. And today I will talk about improved assembly and annotation and first epigenetic map of white spruce. White spruce is an evergreen gymnosperm, which holds great significance as a widely distributed species across North America, from central Alaska to the eastern regions. And its known characteristics include its resilience and adaptability, leading to its description as plastic species, as it can repopulate areas at the end of glaciation. And it's showing ability to grow under challenging conditions and highly variable environments. But white spruce faces several challenges that can hinder its growth, one of which is spruce budworm. It poses a significant threat for the spruces, weakening and even killing trees, causing severe defoliation across over tens of millions of hectares during outbreaks that can last for several years. White spruce forests are also highly susceptible to fire. Intense and frequent fires can lead to significant losses of white spruce stands, resulting in reduced abundance and decreased population density. And white spruce also holds significant economic value, primarily driven by timber harvesting. The species' durable and high-quality wood is widely utilized in construction, furniture production, and paper production. So genomic studies on white spruce can be crucial, as they can provide insights into genetic diversity and adaptive traits of the species. They can help research the impacts of climate change on white spruce populations, and they can contribute developing effective conservation strategies and sustainable management practices to maintain long-term viability of white spruce populations. So as for current genomic resources white spruce, there are two assemblies available, both of which were constructed by B-Roll Lab using short reads. And here you can see some of the metrics that we use to assess the quality of the assembly, one of which is number of scaffolds, which basically tells you how fragmented your assembly is. So lower this number, it is better. And these two versions are similar in terms of number of scaffolds. Version 1 has 2.6 million, and version 2 has 2.4 million scaffolds. And NG50 length is also another metric that we use. So it means half the contigs of this length and longer make up the 50% of the assembly. So higher this number is better. It means your assembly is more contiguous. And this number went up from around 40,000 base pairs from version 1 to 131,000 base pairs in version 2. Another metric we use is reconstruction, which is the total assembly size, which is similar. They're not exactly the same for both of the spruces, but they're similar, which is 21.6 gigabase pairs. And another metric that we use is BUSCO completeness. BUSCO is an acronym for benchmarking universal single copy orthologs, which are a set of highly conserved genes that we expect to detect on the assembly. And by comparing the presence and integrity of these genes, we measure the completeness and accuracy of the assembly. So even though these two versions were great advances at the time, we thought there was some room for improvement, as they were falling short on some of the metrics, especially in terms of number of scaffolds. So we decided to proceed with long reads. And in my project, I'm working with a new version of the white spruce. The aim here is to improve the previous assembly to get more improved quality and a more contiguous assembly. I'm using data from Oxford Nanopore Technologies with 49x coverage. And in addition to this long reads, I am also using short reads, paired end reads, and chromium blink reads at the different steps of the assembly. And you can see the assembly pipeline here, and most of the tools that we used on the pipeline are also developed by B-Roll Lab. So if you're working with assembly, you might want to check our website. And on this new version, on this new assembly, the number of scaffolds decreased significantly to 100,000 scaffolds. NG50 length went up to 2.3 million base pairs, which is a great improvement because it means that we're now at the megabase range. and the Biscuit completeness also went up to 56%. So on this final assembly, for structural annotation, we used breaker to detect the locations of the genes on the assembly, and functional annotation was done using NTAP, using data from various databases, including RefSeq, SwissProt, EGNOG, and InterProScan. So the total number of genes annotated for this assembly after structural annotation was around 93,000 genes. But after applying these filters using AGAT to get a high quality gene set, this number goes down to 68,000 genes, which is still a great improvement because in version 2 we had around 30,000 genes annotated, so number of genes increased more than double. And average gene length here is around 18,000 base pairs, and average intron length is 6,000 base pairs. And repeat analysis done using repeat masker revealed that 89% of the genome was made up by repeats, and 56% of the repeats were long terminal repeats. So after building this new assembly, the next step was to compare the evolutionary relationships between different spruce species. So for this step, I used a whole nuclear genome data of the spruces. I estimated the distance matrix using Abyss Bloom, and then I constructed the tree using neighbor joining. You can see that white spruce appears closely related to interior spruce, Engelmann spruce, and Sitka spruce, which is expected because we know that interior spruce is a result of inbreeding between these three spruce species. And we can see that pines are outliers on the tree. They form a different clade, which is also consistent with what we expect to see. After building this tree, I used it in OrthoFinder to identify the hierarchical orthogroups between white and other spruce species. Here at this step, I filtered for the genes that are longer than 1,000 base pairs long to filter out the potential artifacts and false positives. And after filtering, I had around 42,000 genes. And of these 42,000 genes, 38,000 were in orthogroups, and 2,000 of these genes were in white spruce specific orthogroups. So using these 2,000 genes, I performed Go enrichment analysis. The aim here was to determine whether certain gene ontology terms were over or underrepresented within the gene sort of interest, which is the white spruce specific genes versus the whole gene set from other spruces. And here you can see the most significant term is the green leaf volatile biosynthetic process. Green leaf volatiles are emitted by green plants under biotic stress and are involved in response to mechanisms against herbivores and pathogens. They can attract predators or parasites of herbivores. They can repel herbivores directly, or they can signal neighboring plants to activate their defense mechanism. And they're also involved in a response to abiotic stresses such as heat or drought to control water loss. And another enriched term here is defense response to bacterium incompatible interaction, during which the plant resistance can trigger a hypersensitive response. And hypersensitive response is defined as rapid and localized cell death that occurs at the point of pathogen penetration to avoid the spread of infection to the other areas. And this targeted defense mechanism allows plants to allocate their resources efficiently and prioritize their response to different threats. So moving on to epigenetic map, I also worked with methylation data that comes with nanopore sequencing. And the first initial analysis of the nanopore reads were done by Wahid, who is another PhD student in Dr. Steve Jones' lab at Genome Sciences Center at BC Cancer. And after his initial analysis, we detected 320 million CpG sites on the assembly. And 120 million of these CpGs were fully methylated, which makes up around 37% of the whole CpGs. And the total number of CPGs on genes were around 23 million, number of CPGs on exons were around 2 million, and number of CPGs on introns were around 19 million. And here I plotted the methylation levels of these CPG sites from start to the end of the different all genes, introns, and exons, and after normalizing for length. And here you can see that the methylation level of introns is higher than both mRNAs and exons. And mRNA methylation levels actually reflects the weighted average between introns and exons. And because introns are longer than the exons and they make up most of the mRNA, the methylation level of mRNAs is closer to the methylation level of introns rather than exons. And the methylation goes down towards the beginning and end of the features. I then plotted the methylation levels of all genes, but I grouped the genes into two groups, genes that overlap with the repeat sequence versus the rest of the genes. And here we can see that I also extended the regions of these genes to kilobase pairs upstream of transcription start site and 2,000 base pairs downstream of the transcription end site. And here we can see that genes that overlap with a repeat have higher methylation levels compared to the genes that don't. Analysis on the methylation data also showed that there are around 12,000 differential methylated regions. And the average region of these lengths is average region length of differentially methylated regions are 1,030 base pairs, and there are around, I think, 1,500 annotated genes that are overlapping with the differentially methylated regions. And when I performed enrichment analysis on these genes, it shows enrichment in genes with code terms related to transcription, regulation, and response to wounding. So in conclusion, an updated version of the assembly and annotation is available for White Spruce, and it will be publicly available soon. I'm working on it. And the analysis of the structural and functional annotations reveal genes enriched in defense mechanisms. And also first epigenetic map of the White Spruce is also available with 320 million CPG sites and 12,000 differential methylated regions on 2 haplotypes. I would like to thank my lab members for their support and feedback. And I'd like to thank you all for listening to me. And if you have any questions, I'd be happy to answer them now.
We have time for questions. It was not needed the warning of 1 and 3 minutes. Questions? Yes.
Have you looked at all at which areas of the genome are reduced in your version 3? Because it looked like the total assembly size was about a gigabase smaller than the previous two.
Mm-hmm. I think the total assembly size changes because it's the sum of the length of the contigs, and some of the scaffolds that we had for the previous versions are actually redundant, and they come from different alleles, so they don't actually reflect the true size. So I think, and actually the pipeline for the, I cannot go back, but like the pipeline also that we used for the assembly was different. So for previous versions, we used short reads, and we used a different tool. We used, I think, Abyss for that different versions. And for the new version, we used long reads and we used Shasta. So that's a big difference between the main component of the assembly. So I think that's the reason why we see a difference between reconstruction size.
Do you want to go backwards?
I tried to go back, but it's all right. Yeah, got it. But it's all right, yeah. I was going to show you the pipeline. Yeah. The pipeline, we used Shasta, and we used long reads for this one and short reads for the previous one.
Yes.
Do you have any plans for this epigenetic map? What do you plan to do with it?
Epigenetic map? We actually don't have any. We do have plans, but what I was going to say is that we don't have the transcriptome data yet for this species, but for this genotype. But there are some publicly available RNA sequences data. So we were thinking maybe we can take the analysis next step further with this using this transcriptome data. I think we can use that with that.
Yes, go ahead.
You reported, I think, about 60% BUSCO score, but almost 70,000 genes.
Oops.
Many more genes than lots of other. Thank you. That's many more genes than lots of other plants, but not a super high BUSCO score. Do we expect the BUSCO score to be that high? Yeah.
I think the reason that the BUSCO score is not that high is because the BUSCO genes set that we use for White Spruce is not White Spruce specific. I think we're using embryophyte set, so they're not 100% match. But as long as I remember, for White Spruces, the percentage is around 80%, and highest BUSCO completeness was, I think, for red cedar, which is around 86%. So it can be higher, but we don't expect to see 100% match.
Maybe the follow-up to that, of your 70,000 genes, how many of those do you think are real?
I would say a big percentage of them is real because we applied many filtering here to get the good quality, and we also filtered for the functional annotation, so we just chose the genes that we could map the functional annotation to. So I think the large percentage of these genes are real. And because we use long reads, we have like high confidence in them.
We have time for other questions? Even 2 questions. No questions? So, yes, go ahead. Go ahead.
So, the number of annotated genes that you have, do you have any idea what number of those are duplicated genes?
I'm actually not sure, but during our analysis, like for after structural annotation, I performed filtering for the genes that we could annotate it, and I removed some of the genes were duplicated because of the breaker. It's tool's fault mostly, and I can say that I filtered out the genes that had the same start and end position. So there were some duplicates, and I think this number is the one is actually reflects the true gene. So it's after the removal of duplication. So yeah, I think it's true.
Okay, thank you very much. We finished this session. Thanks, Irem. And
thank you.
There are some announcements.
So anyone that is planning to go on the post-conference tour tomorrow, if you've signed up and you're actually going to come with us, we're going to have an organizational meeting in the middle lunchroom here, just right now on the break. So come on over there right now. We'll do a quick meeting. Otherwise, it's break time, and we're back at 2:40. Thank you.
Thank you, too.